Chara. It is one type of algae belongs to chlorophyceae class. See, we discussed about the Chara classification. Classification of Chara is division Thallophyta, subdivision Algae, class Chlorophyceae, order Corales, family Coraceae, and the genus is Chara. See, we know what is the main character. What are the main characters of the Thallophyta? The plant body cannot differentiate it into root, shoot, and leaves. That plants are placed in the division of Thallophyta. Then under Thallophyta division, they divided into subdivision. Here it is the subdivision of algae. The thallus is green in color. The green color thallus are placed in the subdivision of algae. Class Chlorophyceae. That green color algae are contains chlorophyll A and chlorophyll B pigments. So they are dark green in color. And order is Corales, family Coraceae, genus Chara. It is the true plant Chara. Mainly the Chara plants developed in fresh aquatic environment. Fresh aquatic environment. They are look like a horse tail like structure. So that horse tails are developed in the aquatic region. So these horse tails are also called aquatic horse tails. See, terrestrial horse tails also there. Equisetum. Equisetum is a look like a horse tail like structure, but that horse tail developed on the terrestrial region. That equisetum plants are called terrestrial horse tails. But here Chara look like a horse tail like structure. These horse tail like structures are developed in aquatic region. So these are called aquatic horse tails. The surface of these plant body covered with the silica chemical. When we touch the Chara plant surface, it rough to touch by the addition of silica chemical at the peripheral region of the cell wall. So these are also called stonewarts. Stonewarts means the Chara surface covered with silica chemical, it rough to touch like a stone like structure. Now it is the Chara plant. Chara plant mainly shows two major parts. One is rhizome. See, rhizome is creeping on the soil surface. On this rhizome, aerial shoots are developed. It is the main axis. The main shoot shows nodes and internodes. Sometimes from each node, lateral branches are developed. The lateral branches are in unlimited growth. The lateral branches are also called lateral shoots. The lateral shoots are in unlimited growth. So these branches are called unlimited branches. Unlimited in growth. So these branches are called unlimited branches, are also called shoot of unlimited growth. At the same time, the unlimited branch also contains nodes and internodes. From each node, limited branches are developed. That limited branches are limited in growth. They are green in color. That limited branches also shows nodes and internodes. See, these are the nodes and internodes. From these nodes, small scales like structures are developed. They called secondary laterals. They called secondary laterals. On the primary axis or on the main axis, limited branches also developed. That limited branches are also called primary laterals. Primary lateral means that limited branches developed on the main axis called primary laterals. That primary laterals also containing nodes and internodes. on this node small scales like structures. They called secondary laterals. If the primary axis or main axis produce lateral branches, unlimited branches. They are also green color, unlimited branches, main axis, primary laterals. These all parts are green in color. They are participated in the photosynthesis to prepare food material. A lower part of these rhizome or main axis, rhizoids are present. The rhizoids are mainly helps in fixing of the plant, fixing of the thallus on the substrate, and also helps in the absorption of minerals and water from the soil. It is the main external structure of the Chara thallus. Now we discussed about the Chara cell structure. Chara cells are tube-like structures. They are mainly in rectangular shape. The rectangular shaped cells of Chara containing perfect cell wall. The cell wall mainly made with cellulose, hemicellulose chemical. But outside of these cell wall, silica chemical is present. When the formation of silica chemical at outside of the cell wall, it gives a rough surface to the thallus. So they are called stoneworts. Below the cell wall, a thin plasma layer is present. The plasma layer is also called plasma membrane. The plasma membrane mainly made with phospholipids and protein chemicals. It is in semi-permeable condition. And below the plasma membrane, protoplast is present. The protoplast divided into two types of layers. They are exoplasm and endoplasm. Exoplasm means outside or below the cell wall, liquid juice-like structure is present. In this liquid juice-like structure, cell organelles are absent. Only liquid juice-like structure is present. That liquid juice-like structure area is called exoplasm. Below the exoplasm, endoplasm is present. The endoplasm contains cytoplasm, including all cell organelles. Between the endoplasm and exoplasm, a thin layer is present. The thin layer is called exoendoplasm layer. Exoendoplasm layer. Below this exoendoplasm layer, endoplasm is present. In this endoplasm, all cell organelles are present in the cytoplasm, mainly nucleus. Nucleus is the major part of endoplasm. It is in haploid condition. The cell all activities under control of the nucleus. Near this nucleus, in the cytoplasm, at center is a large vacuole is present. The vacuole acts as a excretory organ of the cell. It protected by thin layer. The thin layer is called tonoplast. Tonoplast is a protective layer of vacuole. That vacuole is also called central vacuole. And in the cytoplasm, mitochondrions, chloroplast, ribosomes, endoplasmic reticulum, Golgi operators. These all are present in the endoplasm. And one more important point in the endoplasm, the cytoplasm or protoplast moves around the vacuole. They are moved by the descending stream and ascending stream of movement of endoplasm. Descending stream means the complete endoplasm moves to downside, and ascending means the movement of protoplast or endoplasm moves to upside. They are moves to upside and moves to downside around the vacuole. These movements are mainly helps in spreading of nutrients, minerals to total cell. It is the movement of endoplasm. And main function of mitochondrion means generally in advanced plants we called mitochondria. In mitochondrion word is used, it is the primitive mitochondrion, the ancestor of the mitochondria. The main function of mitochondria is synthesis of ATPs by the aerobic respiration. And the chloroplast containing chlorophyll A and chlorophyll B pigments, they are mainly helps in preparation of food by the photosynthesis. And remaining endoplasmic reticulum, like rough endoplasmic reticulum, smooth endoplasmic reticulum, ribosomes, Golgi operators, these all are present in the endoplasm. It is the Cara cell structure. Now reproduction, the Cara reproduction mainly done by the two ways. One is vegetative reproduction, second one is sexual reproduction. Asexual reproduction is absent in this Cara. So vegetative reproduction also play a major role in the life cycle. The asexual reproduction done by the bulbils, tubers and amylum stars. See, tubers means on the node area of the major shoot or primary shoot, starch layers, starch cells are formed as a tuber-like structure. If the tubers are leaf from the parental Cara plant, they can develop into new Cara. It done by the tubers. Bulbils means the small size of starch particles. They are also developed on the node area of the main axis. The bulbils also leave from the main shoot or main plant. They can also develop into new Cara plant. And amylum stars. See, amylum nothing but starch. Starch like stars also developed. They are developed into new young Cara plant. The amylum stars are developed on the node area of the main axis. See, on the main axis of the main plant, tubers are developed, bulbils are developed, amylum stars are developed. These all parts are helps in the development of new young Cara plants. This reproduction is also called vegetative reproduction of Cara.